I think the purpose of today's meeting is for us to explain to you about funding when as groups you raise money from your bake sales, your marathons, and the other incredible things you all do, where to put it, and where is it best spent, and what are some of the problems you have. So I thought what we'd do today with these other speakers, we'd talk about first patient groups and how to get together to join things. A little bit of what you've already heard. I must say there's been a little nice overlap. And then we'll talk about basic scientists and how important that is, but how difficult it is sometimes to work with basic scientists, and then how the basic science infiltrates its way into drug companies for the development of the therapies that you need. So let me let the panel introduce themselves. First of all, Deborah, would you like to introduce yourself?
Sure, my name's Deb Garrity, and I come from Sidon Development. We're an orphan drug accelerator, and I'll get into more details. Do you want me to start or just pass it along first?
Just introduction
Sure.
To start with.
Hello, everyone. I'm really pleased to be here. I'm James McLaughlin. I'm from Third Rock Ventures in Boston.
I'm David Pierce from Sanford Research.
I'm Ethan Perlstein. I'm an independent scientist based in the Bay Area.
Okay, good. I have to have one of these microphones. Okay, well, let's start talking about how groups of patients working with and foundations can work together. And I'm going to ask Deborah to introduce that topic, if that's okay with you.
Sure.
Okay.
And I'll start with just a quick sort of bring that back to what Sidon Development is. We're an orphan drug accelerator, and I'll explain what that means in a few minutes, but we saw a lot of room in the rare disease space for a new model to do drug development. And in April of this year, we launched the company with financial backing from NEA, Pfizer Ventures, and Alexandria Real Estate, who also saw that there was a big unmet need in getting projects and getting products from that translational period after having good animal data and getting it into the clinic. So what does Orphan Drug Accelerator mean? I guess a little bit what I can do is say that, give you a little bit of insight of where the name came from. Sidon is actually a derivation of an Old English word that means to reveal. And what that means is revealing new opportunities, new development paths for different products, revealing new collaborations. And Sidon is meant to be sort of a new model of working with foundations, advocacy groups, patients, physicians, so that we can help match products with those patients and get products through that translational period as quickly and capital efficiently as possible.
So I guess I can introduce a little bit about what Third Rock does, and specifically in the rare disease space. So we are a firm that builds companies from the ground up. We've built 31 companies in the last 6 years. And initially, the goal in the rare disease space was to develop companies around really innovative areas of technology and science, really breakthrough therapies that can make a dramatic difference for patients. Initially, the goal was one company every year. In the rare disease space. And we launched a couple of companies, including in the early days, a company called Bluebird Bio, which recently went public, a company called Edimer, a company called Lotus Tissue Repair. And over time, what we've learned is actually we have more companies in the rare disease space than we planned. And the reason for that is when you invest a lot of capital in really interesting areas of biology, what you end up doing is learning more about pathways, more about science, and you learn more about molecules that can really help specific populations. And now we have 9 companies that are working in rare disease space out of about 31, and they're developing therapies that are rapidly going towards the clinic. Overall in the portfolio, we've got 22 products that we brought to the clinic in the last 6 years, when you think in aggregate of 31 companies. So we are, I guess, really dedicated to rare diseases. There are more rare diseases than we can invest in one at a time, but it's something that we really believe in.
So I'm one of those geeky scientists that's been on a journey in the last few years where some of that geeky science translated to the first ever clinical trial for a disease called juvenile Batten disease. So looping that round to the question, and how can you work with scientists, and how can you actually work with different companies in drug development? It's going to be different for every group. It's going to be different for every disease. I can give you examples of how we got to a clinical trial because we first built a registry, and then we first built an actual case study because we knew that we were going to get some scientific findings that would relate to that disease. But an important part of that was we built the registry with a natural history study by approaching physicians that were totally unfamiliar with the disease that I was working on. So getting an outside opinion on anything you do is really very important, in my opinion, because there are so many inherent biases in research, particularly in basic science, but now in translational science. So how do you work with individuals is seek opinions. There's lots of people who will volunteer their time and be intrigued by what you're actually, looking to do. And then, of course, in terms of the actual specifics of drug development right now, there are lots of ways in terms of screening different drug candidates. But I would emphasize that when people bring these ideas to you, think about the ultimate thing that you need right now, and it's got to be something that's probably going to be easy to get through the FDA. And there's a lot of drugs out there, a lot of orphan drugs that are FDA approved.
So I come to the rare disease research space from the basic scientist perspective. I was in academia for many years, but then, like a lot of people in my situation, came up against a very difficult job market and a dwindling federal research budget for the sciences. And so I decided to strike out on my own to become an independent scientist. And I arrived in the rare disease space kind of through a combination of both personal reasons, a family member being diagnosed with a rare disease, but also realizing in my own teaching about genetics that I think a big problem with rare disease research is that we've forgotten about genetics. And so I'm very interested in revitalizing and innovating at the earliest stages of drug discovery in terms of creating personalized disease models. And I think that I represent the tip of an iceberg of professionally trained scientists who may not be able to be absorbed by academia, may not be able to be absorbed by pharma, which is still downsizing, but maybe they're realizing they're tired of just doing science to get a publication or doing science for some other reason. Maybe they want to do their science because it matters. And I think that being a mission-driven scientist in the rare disease space is hopefully something that's coming, and I would like to add that part to this perspective.
Thank you. For myself, I got interested in rare disease about 2008 and started a Center for Rare Disease Therapies. And I was very fortunate to meet the director of the Office of Orphan Products at the FDA, Tim Cote. And Tim Cote came and visited my center, and we ran a workshop for students that just like a 3-hour workshop for them to write an orphan drug designation. And lo and behold, they all did it. So we said, well, if our students could do it in 3 hours, maybe we could have companies come here and do it in 3 days. So that's what we did. And we had several workshops, and these workshops are now in one form or another going around the country. And actually, Chris Hempel, who I see over there, she was one of our customers and successfully got one of her ideas as an orphan drug designation. So we're very, very happy about that. And just recently, Sue, you're here? Yes, we have in common that one of our students, actually my PhD student, wrote a successful orphan drug designation for Tay-Sachs disease and for Sandhoff disease. So I'll come back to that, but I think it's open to lots of people to be able to get a designation and then find other resources to develop the drug. So let me just keep the circle going on different issues, and I'll hand it back to Deborah.
I'll expand a little bit on Sidon's model, just because I think it applies very well to this particular forum. One of the things, as we went out and talked to both investors and then went out after launch and talked to different advocacy groups and foundations, was that there was a real desire to partner with drug development professionals, but also outside of, the traditional biotech and pharma companies. Could they have access to expertise? Could they have access to individuals that had a robust amount of experience getting drugs and making the right decisions on what drugs should make it into the clinic? And I think for it's a pleasure to be here, because I think what we want to do at Sidon is make sure that patient advocacy groups and foundations are empowered, that they feel that they can be part of that process, they can be part of the conversation and have a seat at the table when it comes time to making decisions around trials, engaging with the regulatory authorities, and actually getting the product onto the market and commercializing that. And that then feeds into the research that goes back into the disease. So one of the things that Sidon, it's very important for us, is to continue to have a laser focus on the patients. Who is it that we're serving for Sidon development? And that's the patients, and serving their needs rather than, serving some other. I mean, we have investors, but at the end of the day, your investors are happy because you create successful products that serve patients.
Yeah, I think that's something that I wanted to emphasize too. I don't know if people in industry talk about the purpose of why we go to work every day enough, and we go to work because we are really excited to help patients, and really that's what drives us. And there are a lot of problems, technical hurdles that stand in our way, but I think everyone comes into work in the morning and they're driven by that. And typically, one of the first things I think about when I look at a new therapy for a new rare disease that I don't know very well is, wow, really be fantastic if we could help these patients in some way. So I think that's important to emphasize. On the translational side, I mean, I was thinking back to the conversation we were having about registries, which was a great conversation, a really fantastic conversation. And the great thing about registries and natural history data is it really does help drug development on the clinical side. There are a number of foundational things in addition to that are also extremely helpful. And we haven't really talked about them very much yet, but that includes things like disease models, animals that have symptoms that look like the patients, both mouse models and large animal models. Things like being able to measure critical proteins in blood of patients to measure if the drug's having an effect or not, and functional clinical endpoints that you can actually measure and reproduce. Overall, I think one of the fundamental problems in bringing therapies to the clinic is how unreliable experiments are kind of across the board. And people don't often talk about this, and there are different papers, and probably other people on the panel can quote different numbers, but probably the minimum number of experiments that can't be reproduced is 66%. That means when you see an experiment, it's likely not to be true when you redo the experiment. And I think everyone's probably encountered that before. That's part of the reason why therapies don't work so often and why they're toxic so often, is because we can't reproduce experiments in the right way, and there are a lot of methodological problems with the way that we do experiments. So that's why we at Third Rock, and I think as drug developers overall, spend a lot of time focusing on things like how good is the animal model? Do we have a biomarker? How exactly does the drug work in the cell? And the reason for that is because we don't want the drugs to fail. And we don't want them to have the situation where they look spectacular in one paper and one experiment, but they don't work in future experiments. So I think it's really important to focus on some of those questions. And also, there are things I think the patient advocacy communities can do to make these experiments more reliable. Some of the communities that have decades of R&D, I think there were people from the DMD community, for example. Some of the older communities, Tay-Sachs Foundation, some of the older communities have much better validated preclinical tools that make it easier to bring therapies to market. And it's something that I think a lot of people in this room could add a whole lot of value to in reducing some of the risk in translation and development.
Well, I think there's not really much that I can add other than that obviously it's very complicated working on a rare disease. And But we shouldn't give up. I think the only thing I'd like to add, because I know when we've talked about other things that we want to cover as a panel, the thing that I would just like to emphasize, one of the other hats I wear is I oversee the research entity for Sanford Health, which is the largest rural healthcare company in the United States, which is a non-for-profit. And what I've learned in that role is that, from their perspective is they're a healthcare organization. We have a very large health plan of our own, and they make it real in terms of taking some of those ideas and taking some of that science. It's not translational, it's translatability. Can you actually take that type of research and apply it to your patients? And in the context of today, the patients being individuals with a rare disease. So you have to be very selective and very, very selective with some of the science. Every one of us scientists thinks we have the best idea, but it has to go through some sort of selective process, and it has to be real and truly applied to the disease that you want to apply it to.
So I was really struck this morning by how when patient advocacy groups want to, they can do amazing things. They're capable of amazing fundraising feats, amazing feats of marketing and awareness. And I tweeted, because I'm a Twitter addict, this morning in the session, that what happens when patient advocacy groups start applying this same zeal and passion to patient-driven drug discovery or patient-driven clinical trials, all the aspects of this R&D pipeline that have normally been done by somebody else, by a partnership between pharma and academia. And I want to tell you that there are actually examples of this right now. So there's an example of a woman who found out that she has the mutation for rare familial insomnia, which is a prion disease. And she got tested and found out that she has this mutation. And so she decided that she was going to change her life. And so what she's doing now is actually crowdfunding I guess what you could call maybe the first step toward a cure. And what she's done is essentially take an experimental compound was published on by some academic lab, but it's languishing in that person's freezer, or no one's done anything with it, as often happens in academia, and has combined that group with another group that has a mouse model for this particular disease. And so they've only raised $9,000 so far, but it's an example, I think, of what happens when patients start driving this process. And it's possible to do it at the drug discovery side, but it's also possible to do it at the very other end, which is the clinical trial phase. So I don't know if you're aware, but there's a rare disease called black bone disease, alkaptonuria, and they're right now actually doing what appears to be the first patient-driven clinical trial. And so they want to raise $98,000 on Indiegogo, which is like Kickstarter, one of these sites, and they've already raised almost $40,000 in the last 3 weeks, which I think is a pretty impressive feat as someone who watches a lot of science crowdfunding. And so I think that the perspective I would just inject is that there are these traditional structures in place that are clearly modernizing and keeping up with the times to some degree, but I think there's nothing like, as was said earlier, the innovation that's catalyzed by the rare disease patients and their advocates. And I think there are now living and breathing examples, thanks to the democratizing potential of the internet, that shows that patients and their advocates can take over from the earliest stages of drug discovery and even potentially shepherd through a compound or some therapy all the way through to the finish line.
You may be wondering what you can do. So one of the things that we found is that the FDA has changed since last year with this fiducia rules. They're very open to hearing from patients, because patients, or patient advocate groups, because what the end point should be. If you're going to have a drug, what would be a huge improvement over what you have. And what drugs don't work very well and where they could be improved. So the FDA has actually set up an office of specific health that they want to, and they have a professional who's in charge of that, who actually want to hear from you. They actually want to hear at their next meeting, whenever that is, of different diseases, they want to hear what you think and what endpoints you would recommend to them. So that's one way clearly you can get involved. Now, I don't know if anybody's here from the Cystic Fibrosis Foundation, but the Cystic Fibrosis Foundation is probably the singular most astonishing group. They raised $75 million and paid for their own clinical trials, and that has resulted now in a new drug that has been developed with a company and it's a kind of a gene therapy approach, and it's approach of the future, and it's apparently very effective. So it can be done. Now, of course, it took years and years to raise that kind of money and that kind of support, but that's it. For those of you who have like a two-person foundation, you're at the beginning. You never know where this is going to go. The other thing is there's some crossover between rare diseases. I mean, most of them have ridiculously unpronounceable names, but if you get over that, you find that there are common links. There's a whole area of rare diseases of lipid storage or lysosomal storage deficiency, and within those groups, they're very famous names, and we've already mentioned a few. They are probably all related genetically. They're probably all related in one way or another to the kind of therapies that you can bring to them. And if we switch a little bit now to therapies, I'm very excited about the potential for gene therapy. There's been a real tragedy in gene therapy. A few years ago, a young man, 17 years of age, was in a trial that should never have happened, was treated with the wrong vector, the wrong delivery of the gene therapy, and died. And he was a martyr to it. And all gene therapy work stopped. And then people started, particularly at my old university, University of Florida, to test gene therapy in a blind dog. And that, you know what it is like when you don't have a friend, you get a dog. And this dog, a French Briard, is born blind at birth. And they injected with a vector called AAV, adeno-associated virus, which is very safe and doesn't do any harm, and they injected into that eye the missing gene that he was born with. And then he began to see from one eye. And actually, it was just fantastic. I mean, you hold a pencil here and he'd look. You hold a pencil here and he wouldn't look because the other eye was still blind. And that dog went to Congress. And all the congressmen got out their pencils and were going one side to the other. And if ever you needed a lobbyist, take a dog. I mean, that was the best. So that disease, which has now been tested in lots of people for lots of years now, and has actually kind of biblically brought sight to these patients suffering from labus amaurosis congenitalis, they are seeing. And so that's first really gene therapy that's really working very well. And they're doing it very carefully. As I say, they did it a few years ago. They're looking for any problems. They've started to do the second eye because they only did one eye to begin with. So I think that kind of vector and that kind of approach is going to make a big difference in all the diseases. When I was talking with my colleagues at the lunch about hemophilia, it could be used for hemophilia too. It could actually provide a cure. Now, the only trouble with a cure is the drug companies won't know how to charge for a cure. And that's a subject of another discussion. But I am just full of optimism about what's happening in the science field and what's happening with all the possibilities for rare disease. You want to pick up from there or go forward?
Yeah. If I have two dogs, does that make me a ginormous loser then? I just to go forward on the AAV, I think that gene therapy is, as you said, but I'll say it a little bit differently, it's a little bit in a renaissance. But I think it came from a groundswell because I think people sort of assumed that it was going to be a hurdle that was way too difficult to overcome. But I think somebody took a chance on, and to just talk a little bit about what James mentioned, took a chance on using a good animal model to bring science back into it, and picking up on my colleague down at the end, took it back to the scientific level and took it back to what are the questions that we're really answering for rare diseases. And that's a little bit what Sidon is that we want to ask the critical questions and answer those critical questions for moving a product forward into the clinic. And can you define what those are, fund those in a very efficient way, partner with different foundations, different advocacy groups, and move those forward. You mentioned earlier what can advocacy and the foundations do. ask questions. Reach out to industry, reach out to academia, reach out to colleagues, because we also have questions, and we want to be able to talk to the foundations. You know a lot more about the diseases than we do. Maybe not everybody admits that, but that's the truth, because we don't live with it every day. And I think for me, as somebody who founded this company, the goal is to create the drugs that are going to be the best to address what patients need.
Yeah, so I think that's a great point. I love the analogy of the Leber's congenital amaurosis dog. I mean, I think the important point in drug development often is successful clinical trials are built on the back of decades of research and people pushing and working with basic scientists on understanding the disease a little bit better, understanding little details about how to measure the disease and how to study the disease in laboratories. And when that reaches a critical mass, dramatic things are possible. And I think that's definitely something that the LCA story shows. It's also sometimes a missed opportunity. I'll tell you an anecdote. We were looking at a rare form of congenital muscular dystrophy recently, and we were very excited because we found a report in the literature that there was a dog with this exact same form of congenital muscular dystrophy. And this would mean that we could put the investigational drug in the dog and have a pretty good idea about how the protein moved throughout the dog's body, and it would give us a good idea about things like dose and things like toxicity to take it forward to the clinic. And we looked at this paper, and we talked to the researcher, and the researcher said, this was an interesting dog, but the dog actually, passed away, and we didn't breed up the dog. The dog's genotype was lost. And that was an opportunity that was lost. The patient group could have come in and bred the dog and had a colony, which would have supported research in that disease for a long time. It would have made a big difference. And they didn't do that, and the dog was lost. But the good thing is those serendipitous things happen all the time. One of my colleagues was walking down the street in Boston, near our offices on Newbury Street in Boston, and saw a dog with ALS, and that was a dog that hadn't been characterized before, a genotype of a dog that hadn't been characterized before. So opportunities like that come along. I think the critical thing is thinking about some of these foundational elements of science that can really help take therapies forward. It's not just information on the clinical population. It's also information on some of these tools that really make translation possible, make it easier to take therapies forward into the clinic.
So I'm really worried now. I have an energetic black Labrador that wouldn't look at a pencil if I put it in front of it. Probably just chew it. So again, just to reiterate, I think the one thing I would just like to circle back on, whether it's gene therapy, drug development, or whatever, many times that we are talking about symptomatic treatments, and I'd like to reiterate what was said this morning about registries. It's perfectly fine to have a disease-specific registry, but I can think of dozens of diseases that have untreatable seizures right now, and as we develop particular targeted therapies for seizures in one specific disorder, the underlying basis of those seizures in another disease could well be the same, because these are secondary consequences of the disease. So pooling everything in a national registry, and we have one as that Liz Donahue here, some of runs through Sanford, I think is very important. And again, when you can link that to a medical record as well, which I know not everybody has, is very important. But just circling back to the large animals, absolutely, we need to develop better animals. Mice are really stupid when it comes to a lot of experiments, and so are us as scientists, because we haven't recognized the fact that when it comes to looking at the brain of a mouse is the size of my thumbnail. So that's not really going to be the solution for us when we want to do gene therapy in the brain. The eye is a wonderful story. It's the most accessible organ that we have. So we need to develop better models, and serendipitously, there's the dogs, there are sheep. There's a new realm coming along now. We can genetically modify pigs, and in the next couple of years, you'll hear more and more about the use of pigs. The cystic fibrosis model for the pig has been well published and well studied by Mike Welsh at the University of Iowa, And the technology that's been used behind that is coming down in price. And if you check the NIH website, you'll see there are a number of SBIR grants where pig models are being bred up for different rare diseases right now. And not just for the brain, but for the physiology of human beings, these models will become much more appropriate for many studies.
So I'd just like to briefly expand on the point about disease models. So whether people know it or not, there actually is what I call, and other people have called, a mouse monoculture of disease models. In other words, basically, if you want to model a disease, it's basically mice. It's there's evolutionary reasons why you would use mice as a surrogate or a proxy for people, but it come to a point where now that's essentially the almost the only organism people use to do the basic kind of discovery science that allows you to understand what's actually wrong at the molecular level. So I would actually like to advocate not even small animals, but non-mammals and maybe non-vertebrates. So, for example, my favorite case is, so I'm specializing in the lysosomal storage disorder. So the case I like to bring up is of a disease called Niemann-Pick type C. So the gene responsible for the gene that when mutated is responsible for 95% of NPC cases is a gene called NPC1. Turns out the NPC1 is an ancient gene. It's 1 billion years old. Now, how do I know that? So you can look at a yeast cell. So the last time that we and yeast shared an ancestor is a great. So 23andMe and yeast, yeast did 23andMe, it would show that we have a long distant cousin. But if you look at the DNA level, and you take the NPC1 gene, which we have, and you look at the yeast genome and ask, is there a gene that looks like that? Turns out there is. And it doesn't stop there. If you look at a worm, which is a little bit more complicated than a yeast, actually has different cells. A yeast is just one cell, but a worm has multiple cell types. You look there, turns out there's a NPC1 version of that. If you look a little bit more complicated at a fly, there's a NPC1 version there. You go all the way to a zebrafish. It's not even a mammal, and you can find an example of this gene. And the zebrafish, 75% of its genes are shared with us. So as much as a mouse is a great proxy for human physiology, I think actually part of the challenge we face in terms of reinvigorating the earliest stages of drug discovery is to have a more expansive view, a kind of polyculture of disease models, not just about mice, but thinking about if the gene is actually ancient. Now, there's certainly some rare diseases where it seems to be a uniquely mammalian gene, right? There's no ancient version of it. But for many of these diseases, there actually is an ancient version. And if you're not studying disease models in that simpler case, I think you're actually missing a huge opportunity to build from the bottom up a really solid foundation of understanding of what's going wrong at the molecular level.
Thank you. I just want to say one word about the basic science, then I come to questions. I have a question here. Basic scientists are a weird bunch of people. And I've been one for a long time. And we do things, just like you're hearing, on weird animals, and we're biologists. We don't really care about a disease. That's the problem. So when you're talking to a scientist, and you're thinking, can this scientist cure my disease? He probably isn't doing anything with the name of your disease. But he may be doing, or she may be doing, very, very vital work in understanding the mechanism of that disease. So if you raise some money and you want to give it for research, don't just look for somebody who can deal with, say, Niemann-Pick's disease. Look for somebody who can deal with the MP1 gene or look a little broader. And that research might very well help your son or your daughter get ultimately a treatment or a cure. Having said that, I have a question coming in. I see some questions from the floor, so I think in the last few minutes we should try to answer questions. This question from Marianne is about as broad a question as we could cover. What are the critical questions imperative for research and drug development for rare diseases? Any takers?
Money. So, I mean, obviously, but use it wisely. So, the panel, we have a panel here, so I think it's time that we disagree. I've published 20-odd papers on a yeast model for Batten disease. I'm not going to find the cure for Batten disease from that yeast model. I am going to find it from the mouse, and I'm going to find it from the pig that I've built. You have to be very careful with the questions that you ask. I am initially a basic scientist, but I've evolved into something else because you learn. Science is about learning. As human beings, we have to learn. about that. So what's the critical question? Every rare disease should probably be treated differently when it comes to the state of the science right now, because we have 24,000 genes that make us up. They're all there for a very specific reason. Taking a simple yeast, you can remove one of those genes and it will kill the yeast. Taking one of those other genes and the yeast won't even tell you anything about it. So there's a complex thing here that we have to understand in terms of integrating different disciplines. So you want to work on Niemann-Pick type C? I'd go and recruit someone who's never worked on Niemann-Pick type C, because they're going to ask different types of questions. I mentioned this before, you have to be smart and ask your questions. You have to be very undiplomatic in this field, which I have a reputation of, because it's just so complicated. It's not going to be simple. If someone's worked on this for so many years, then it's time to move on and find someone who's going to ask different questions.
So I guess the question was, what's the critical imperative question for R&D in rare diseases? And I would say, a simple question that's really hard to answer is what causes the rare disease? And a lot of times we assume that we know the answer to that question because we have a genetically defined disease in many cases, and we know the gene that causes it. But we often don't know other questions about the disease. For example, why are certain cells affected more than others? Why do we have certain symptoms? If every single cell in the body is affected, why is this the way the disease looks, and why is the disease different in this patient versus this other patient? So the underlying mechanism of the disease is something that basic scientists spend a lot of time on. It's actually really critical, because if you know the answer to that question, you can help figure out what the product profile will look like. You can figure out, where the drug needs to go and how much drug you need in certain places and when you actually dose the patient. So it's actually very critical research. And it sometimes seems a little bit far from clinical application when you're looking at, an invertebrate or something. But oftentimes, these questions of cellular mechanism are actually really critical and ultimately lead to successful therapies in some cases in the future.
Thank you. Is there another question? Okay. Yes, sorry. Question at the back. That's you. Wait for the microphone.
You all have touched on sort of a dispersed array of things that a patient organization could be thinking about in terms of how to be strategic in drug development and the drug development pipeline. But I think a lot of us have heard a lot about the fact that the FDA is openly inviting patient organizations in, that there's a huge role now to play in terms of patient organizations inputting on endpoints and understanding how to support biomarkers and being partners with pharma around ensuring that endpoints that are selected are measured effectively and success is more likely. And I think while we've all heard bits and pieces about that, and we would all like to be enormously strategic around the infrastructure pieces like registries and organizing the patient population and so forth to support these different pieces, it would be nice to have a panel like you all talk about specifically what some of those buckets are. How do we think about our role in infrastructure development, whether it's clinical trial readiness or registries or what have you? How do we think about patient organization? How do we think about our role as it relates to this kind of new space with FDA and how we really understand how we advocate, what pieces are important, how we engage the community to do that and so forth? Because I think, at least for me, what I'm trying to understand from this particular panel is, so what does the role look like today and how is it evolving?
Okay. Do you want to speak to that question?
Kill myself getting up, but I just am worried about the environment, and we all need to make sure that I went to a meeting in Boston recently, and the FDA was there, and they're talking about inviting all the patient advocates to the table, and the whole environment is really changing for us. And it's a little bit scary, I have to say, out here, because we're now, suddenly people are listening to us, or suddenly people do care what we have to say. And that's like, after spending 20 years screaming to be heard, and when somebody says, I really want to hear you, you're like, oh, never mind, So it's kind of it's kind of frightening. So I implore all of us here. I think that the tides are turning and the FDA is inviting us all there. So let's remember that they're saying it to us, we are experts. And let's be proud and let's know exactly what we're expert in. So part of the problem is that I feel, you know how pendulums swing to the other side? So the FDA is now inviting all these patient advocates to hike over to the FDA and eat peanut butter with them. And I worry that patient experts are gonna think that they're experts in every disease and every rare disease. So recently there was a meeting on, pulmonary fibrosis, and the patient expert was in asthma. So we have to make sure that we know who we are, and we actually raise our own standards to only I'm not gonna speak for your disorder. And even though we are an aggregate group and we're a loud, rare voice, if we are gonna be treated like scientists, citizen scientists that can make a difference, we have to make sure that we know our place, we know what we're expert in, and not to be seduced just to be everywhere for everybody. I think that's really important. And then just one last thing. I love you guys so much because you care about rare diseases. And I think we can be cheerleaders. A lot of our groups, when I was raising my kids, every vacation we had in the summer was around a laboratory that was studying mice, and they thought they were going to like Maine. They were going to Jackson Labs, And I tricked them throughout their entire lifetime. Niagara Falls, it was like fox chase. I mean, we went to these labs. And I would, take them to a park, but then I would take them to the laboratory. And my daughter would be invited. These laboratory doctors never even saw a person with HPS. And my daughter would be there looking in the microscope, and they'd be showing some molecule. And I taught my daughter to be very interested. She's legally blind, can't see a damn what's in the microscope, but she'd be like, wow.
And there's a question behind. Oh.
Hi, wonderful story, thank you. My name is Deborah Miller. I'm the founder of Cure Duchenne, and I think it might have been mentioned earlier today that we, as a Duchenne community, had a little bit of bad news this morning in terms of a project that actually Cure Duchenne started funding about 10 years ago, and licensed with Prosenza and licensed to GSK, and it did not meet its endpoints today. So I was a little late this morning. I'm dealing with some of that this morning. But I think, we're not giving up on this technology, and we found another company that's working on this and still have high hope. But I would love to hear a reminder from the panelists on how important it is for a community not to put all their hopes in one drug or one company. Because we've seen this before in the Duchenne community with PTC Therapeutics. Today was another reminder that we have to keep the pipeline really, really full. And cystic fibrosis was mentioned today. It's, we look up to them as a company that has been very forward thinking, and they don't rest on their laurels, and they do keep drugs just continually coming in at all different phases of research. So, maybe you could comment on the failure rate for drugs in the different phases. I think we've all heard it before, but I think it's a good reminder to be aware of that.
Well, if I may just start, and I'm sorry I haven't really answered your question, but with drugs, development of drugs like going into a funnel. And all sorts of things, the basic science, drugs, and everything go in that funnel, and it gets tighter and tighter, and more and more drop out as the process goes through, the phase 1, phase 2, phase 3. And it is disappointing. It's enormously disappointing to companies sometimes that the drug that seemed so logical and so good in phase 1 trials and phase 2 didn't work out in phase 3. And it's part of the system. It's part of the business of developing drugs. And it happens, and you're absolutely right. People shouldn't get their hopes pinned too high on any particular drug, even when it comes out, because then you go into what is called phase 4. And in phase 4, you have to report to the FDA any kind of problems that you have. And you've heard, like Vioxx, that Merck had, people started to have heart attacks until it got to a number that the FDA stopped the drug, and it was as a drug that was going to be a blockbuster drug. So for rare diseases, that's probably a little less likely if it's really working, but it is a fact of life. That developing drugs is a very difficult business model. It has a high payoff at the end for the really good ones that get through, but it is a difficult thing. And it's difficult from the science point of view, difficult from the processing point of view, and so on. Even a company that might have a wonderful drug and be selling it fails to make it properly. And this happened with Genzyme, and Genzyme is no longer a big company now. It's a sub-company of another company because they failed to make the drug properly. So many things can happen all the way along. Anybody want to add to that comment? Maybe you.
I mean, just to sort of go on to take that a little bit forward, I think the newest statistics are 1 in 10 drugs make it to the end. So, that's across all indications, across all diseases. I mean, in oncology, it's somewhere less than that. I think the success rate for oncology products is less than 3%. And part of the challenge with that is they're not translatable from phase 2 into phase 3, and oftentimes projects fail in phase 3. You know, I think it's a daunting proposition, and I think that's why, you know, the people at this table, you know, the work that we're trying to do is you make smarter decisions earlier, so you have fewer failures later, which means animal models, maybe non-animal models, yeast, whatever, and it's asking the critical questions that actually best replicate what I think is the human condition of the disease. And we can only know what that is, to come back to sort of this whole group, is by partnering with patient advocacy. And I think one of the things someone out there mentioned was, you know, what is it that the patient advocacy and the foundations can do? We are looking to co-fund projects, work together on the science, work together on translating these projects to the clinic. What does that mean? It could be financially co-funding them. It could be sharing information. It could be working together with different investigators. So, you know, for me, the best thing that, and for Sidon, the best thing that we can do is you have a higher level of collaboration. And I think at the end of the day, you have greater transparency, greater information sharing, and you can make better decisions on products and early stage technologies that are sitting around either on the shelves of academics or the shelves of pharma and biotech companies.
So this was described as the valley of death in drug development because so many things fail when it comes to development drugs. I just want to circle back, taking this question and the previous question, just in terms of the environment and working with the regulatory aspects. And the environment has changed in drug development. The NIH a number of years ago supported, with major funds, drug development programs for screening what they called high throughput drug libraries. And the NIH is now defunding those because, you know what, academic science isn't drug development. We could have the capability of screening 300,000 compounds, but they were just full of toxins and poisons. They may actually rescue a cellular phenotype, but academia doesn't have the ability to do all the toxicological analysis to make that safe into humans. So the landscape is changing there, and the NIH has recognized that. But also, I want to sort of stand up for the FDA a little bit. You know what? If you can go speak to the FDA, they will help you. You know, I've done this. You're developing a drug. You can do something. You need an IND to do a clinical trial. Basically, you need to get the paperwork to say we can put something in human beings. The FDA has a pre-IND process. They'll meet with you and say, you know what, that's a really stupid idea. Don't pursue it. Or they'll tell you, these are the things that we need to do to advance this drug. And to put it into human clinical trials. So I think we need to be aware of that. I've made it very simplistic, of course. But then on the drug development right now, there's a drug library called the Prestwick Library. So as foundations, this Prestwick Library is a couple of thousand of compounds that have been put into humans and are approved by the FDA. They've either worked in some diseases or have failed in some diseases, but they're human safe. If you have a phenotype, a cellular phenotype, you can screen to see if it changes The enzyme activity changes the phenotype in the cells. You've got something ready to go. For a small amount of money, you can try in your animal model, even the yeast model or the fly model, and see if it modifies the phenotype. That's a start. It'll probably only work for a fraction of diseases with that approach, but it's a good investment. And it is very important to have more than one project or drug pipeline on the go, as we just heard.
I would just put a very quick fine point on that, which is that, I think recent estimates show that it's $10 billion per approved drug. And again, 95% of them that's the attrition rate, so most of them are not making it through. But I would say that I think the latest projections or latest numbers are that the majority of drugs that fail are failing in phase 2 and phase 3. Now remember, phase 1 is establishing safety. Phase 2 and phase 3 are establishing efficacy. So what it means is that pharma's gotten pretty good at making safe but generally not effective drugs for the indication that's been designed. Now the silver lining here is, as Steve Groff talked about it, the repurposing initiative. So maybe you had a candidate in phase 2 flop for disease XYZ, but it's safe. So maybe it's time to see if that could work in a different indication. And I think that has at least put some silver lining on the fact that $10 billion per approved drug is just an insanely obscene number.
Hi, I'm Julie Flygar. I'm a narcolepsy advocate, and I just wanted to mention to everyone that narcolepsy is the first rare disease to go through the FDA's Patient-Focused Drug Development Initiative. Our meeting is on Tuesday at FDA. I'll be flying out tomorrow morning to get there. It's just been a really amazing experience working with the FDA in this process. FDA has really been listening, which just as a patient advocate, you go to so many different regulators at different departments, and you feel like people are just sort of half listening or being polite. And I've really had a great experience with the FDA. Not only has it been educating the FDA about narcolepsy, but it's also been educating our narcolepsy community about what the FDA does, what they are capable of, what they're not capable of, and setting appropriate expectations for this process. And I just wanted to let anyone know if they want to talk more about it, if their disease is going to go through the FPF-DDI initiative. We've learned a lot going through the process, and please feel free to approach me and talk more about that. Thank you.
Thank you for that comment, and that probably is as good an answer as you can get to your question. Let me I don't want to end up on a downer, like, oh my God, we'll never get a drug through. The fact is that since, or before 1983, there were only 10 orphan drugs available. Then the Orphan Drug Act of 1983 was signed into law, and since then, we've had developed nearly 400 rare disease therapies. Now, it's not a great number if you say, well, we got 7,000 diseases to go. Actually, probably we'll never treat 7,000 diseases. We'll treat a small number, but at least that act has really improved things. And then last year, they upgraded that act to make it even more accessible to the public. So I think that expression, we're the government, we're here to help you, is actually true in that sense, that the FDA, particularly the Office of Orphan Product Development, is really trying to help this group specifically and wants help from this group. Sorry, you have a question?
I have a question. How do we get some of these meds pushed through faster for use in pediatrics? Because as a parent to a 6-year-old, she's screwed for many years on a lot of these meds that are coming out to be promising and working well in adults, but we have to wait lifetimes, it seems like, to get it approved for pediatrics when her quality of life can be improved now. And I understand it's a safety and we have to go through that, but what as a parent can I do, whether it's going to the FDA or going further, to do? I mean, look at methotrexate used as a chemo drug. They have found great benefits when used for arthritis in kids. And so there's a lot of those crossover. And the second question is, I've seen a lot of drugs coming out that, failed for what its original purpose was, but have been shown to have great benefits, like methotrexate, but aren't going through to help. And as a parent to a 6-year-old who's got more diseases than she does years on earth, I need meds for her to help. And the few that we have found has dramatically improved her quality of life, but we're screwed. And we've been told many times, looking forward, without these meds, she's not going to live past puberty. And that is frustrating, knowing that those meds are there and I can't attain them.
Yeah, it's extremely frustrating. And but on the other hand, I mean, the FDA is there for safety. mean, if you go back to thalidomide years ago, children in Europe grew up without arms and legs because of having drugs that were not properly tested. So there's a sort of flip side to that. But I could understand that particularly as children are growing, and if you have a disease and they're getting worse all the time, it's very, very frustrating. You have to start shouting. You have to, remember the AIDS people when they first got AIDS? They shouted. They ruined every meeting and they got attention for things. You have to do that. You have to get out there and ruin the hearings and get notice and say, this is really, really important to us. So I'll come along with you. Give me a T-shirt. We have one question. I guess this is the last question of the day for this panel. How do we connect a rare disease interest in a specific gene with researchers who might be interested in studying it? Takers?
Can you read it again?
Yeah. How do we connect a rare disease interest in a specific gene with researchers who might be interested in studying? So researchers somewhere working on that gene, and somewhere else somebody finds it in a rare disease. How do we connect?
Well, I'll just say, as I reiterate from before, I think what we've seen is that the patient themselves can simply act as a kind of project manager and bring the pieces together and provide the marketing, essentially provide a lot of the overhead that would never be supplied by the academic side or there would never be an investment from the pharma side. So I think that you have to rely on the idiosyncrasies of a particular person deciding to take that initiative and have the right personal means and circumstance to be able to say, moonlight and take classes on something to know who to contact. But I actually think there needs to be a generic solution to this, which would almost kind of imagine could be like a Craigslist, where you have patient advocacy groups who post, say, I want to hire a lean and mean team of 5 professionally trained scientists who are going to work on my disease. And then you, on the other hand, you have a bunch of, and as I represent, I think a lot of professionally trained scientists who are not able to be absorbed into the traditional system and maybe are able to say, hey, you know what, we could maybe take up the call, but I don't know if there's such a thing that exists yet. But if there needs to be such a platform, I agree, because otherwise you're left to the devices, although these are really resourceful devices of individuals, patients who would be driving this process forward. It'd be nice if there was a generic way where there was just a place where people could go to find each other.
So the person you want to speak to spoke this morning. Steve Groff's office is very open and very contactable. If you want to be connected, Steve's office and the people at the Office of Rare Diseases will connect you with the researchers. Right, Steve? Yeah. And then the other thing is that if it isn't obvious, there are other people within the NIH at the different institutions, whether it's the Institute of Neurological Disorders or Mental Health or Child Health and Disease. Through Google, you'll get there. But I'm pretty certain the Office of Rare Diseases will be able to help connect you that way. Don't be afraid to ask them. They're working for you.
I just want to thank Kay Thomas Pickard for that last question.
We'll do one more here. Just a comment on how to matchmake, if you like, between research and patient advocates and money and all of that. There are, from the technology platform earlier, there are more and more platforms coming online every day. If you look at Microza, they are posting research projects that you can go on and look at and see if there's a link between what you need and what they're doing. So that's crowdsourcing research and crowdfunding research. There are things like Health Tech Hatch, MedStarter. Polywog is funding that Valley of Death companies from a crowdfunding perspective. The Violet Project, you can adopt a drug in particular research areas. So don't look just for the obvious ways to connect, but go out there and take a really broad look around at what's coming out on from an interactive technology perspective, and you'll find greater access, I think, to more and more things every day.
I want to thank the panel. I want to thank the panel. Thank you so much. Thank you.